This first lecture is an introduction to chemical ecology. I'll tell you a little bit about what chemical ecology is and some of the methods which are used in it. So first of all, animals use their sense of smell to assess their environment, and the information that they get from what they smell, it may elicit a positive response, as you see there with a bee flying towards a flower there, which is emitting some sweet scent to attract the pollinator, or conversely, it may elicit a negative response, such as where you see that skunk there emitting some foul defensive odor to make the predators go away. So this is the basic thing about chemical ecology. It's how living organisms use their sense of smell. It's a multidisciplinary subject, so it's at the interface between chemistry, biology, and ecology. So with their sense of smell, that's using the olfactory system to detect the odor molecules, which are called volatiles. These volatile chemicals, that's what's detected by the olfactory system, and so because those are chemicals, there's obviously some chemistry involved in that, but it's a very interesting interdisciplinary subject because it's the biology that makes the chemistry special. So if there's a particular chemical which elicits a response from the insect, then it's of interest, and it gives it some greater meaning because of the effect which is having on the animals. And as you'll see in one of the next topics, which is going to be about pheromones, then sometimes even very minute amounts of some of these particular chemicals can elicit an amazing response with the animals. So it's an interesting topic, and the unifying theme throughout chemical ecology is biological interactions. So we're interested in these chemicals because they're used as a kind of communication between the living organisms. So there's particular signals which may be emitted on purpose to attract a response, and they've co-evolved to have the emitter and the receiver both being adapted, for example, as you might have with the pollination. But then there are other things where the insects or other living organism will just be using something to its own advantage, and so it might not even be to the advantage of the organism which is emitting it. So for example, with a host location cue, then it's a benefit to the receiver to detect that, but it's not of benefit to the emitter, and it may even be detrimental to it if you get a herbivore, for example, that comes along and damages the plant. So this subject, it depends on the context as well as the chemistry. So there's different things will have different meanings in different contexts, and the insects may even learn to respond differently over time. So there could even be learning behaviour which complicates some of these responses. But it's a sensory cue. So you get smells, and it allows the animal to have better information about the environment. And the critical thing about this is that because it's a signal, then it might often work at quite low concentrations. So it's not like something which is having a physiological effect on the organism which is receiving and detecting the smell. It's something which is used as a signal or a cue for it to which triggers certain behaviours which will improve the fitness of the animal. So if it's looking for a host to feed on or a mate to mate with, it helps the animal to find its way around in the world. So underlying all of this is the olfactory system, and I'm going to show here with insects because this is the model which is often used in chemical ecology, so the insect olfaction. So what you have here is the olfactory receptor neurons, which are enclosed in the antennae, the mouth parts, and the legs. Those are the places where you might find them. They're mostly on the antennae. These are the places where the olfactory receptors are located, and when you get a particular molecule which is fitting to the receptor, then there'll be a binding process, and it'll be a specific binding, so that only particular volatiles will trigger the particular receptors. So it's a specific response to particular odors, and once it's the receptor detects a particular chemical, it fires a message to the brain, say, oh, this particular chemical has been detected now, and then the brain will interpret this, and it might process it. So putting together with the environmental context of what other cues it's detecting at the same time, and it'll make sense of it and decide how to respond. And as already mentioned, there can sometimes be learning behaviours. So some particular smells will be hardwired, and it'll elicit a response which will be always the same. Other ones, there'll be more flexibility, and it'll depend on the previous experience of the insect and the way in which it responds. So if you look over on the right a bit more of this slide, then you can see closer in. So inside these sensili, which are very fine hairs on the antennae, then you've got tiny pores through which the molecules will enter, and there'll be olfactory binding proteins which facilitate the transport of the molecule to the receptor, and that takes it to the receptor, and if it's the right shape and the right chemical, it'll bind to it and it'll open a channel, and there'll be a flow of ions through the channel, so that causes depolarization of the neuron, and that triggers the firing of the neuron and the message going to the brain there, which then be processed and will perhaps lead to a behaviour depending on what the particular chemical that's been detected is. So here we are looking at these channels again, and you can see here there's an odour which is represented by these purple hexagons, and it binds to the odour receptor there, the odorant receptor. This is a molecule with which changes the shape of the receptor, and it opens this channel, allowing the calcium ions to flow through from the extracellular space into the intracellular space. It's constructed of two proteins. So there's the orco protein, which is present as a general one, and then there's this odorant receptor protein, which will be specific for the particular chemical. So when it detects the chemical that it's tuned into, then there'll be this flow of calcium ions through the channel, which will lead to this depolarization. And as it says there, the different olfactory receptor neurons are sensitive to different chemicals. The olfactory system, it will provide a simplified representation of the external environment, and the insect will also take in other sensory cues such as sight, and it'll integrate these things to decide what to do. It's crucial for fitness because it allows the organism to find resources that it needs, such as food, mates to mate with. So it's really important for survival and reproduction of these insects and other living organisms. And although it's often thought the insects are quite simple, it's actually, the more you look into it, it's actually some remarkable sophistication with some of these things, as you will see in some of the follow-on topics. I mean, this is just this first talk is just a kind of overview, giving you a bit of background about chemical ecology. But when you see some particular examples of this, there's some pretty neat things happening, and it's perhaps not as simple as might at first be thought with these what these insects are doing. So, having said a bit about what chemical ecology is, and how we're studying the sense of smell and how animals use that. The next thing I'd like to do is to give you some overview of the methods which are used in chemical ecology, because then the next talks will make a bit more sense if you understand some of the methods that are used. So there's a suite of different techniques that are used together, and they're actually very useful to use these in combination, because it allows us to find out what are the critical chemicals, and how do these insects respond to it. But usually the chemical ecology research will start off with collecting some sample of the chemicals which the insect is thought perhaps it'll respond to. So you have some test odor substance, and you need to collect the chemicals, collect the smells. Now, how do you do this? So the technique which is usually used for the volatile collection is headspace sampling, and so this is a system of pumps, and it'll pump in clean air into the vessel. So you have either a glass vessel here that's partially enclosing the test substance, or you can use these PET bags which will do the same thing. So these are special bags which you put in the oven before to remove any contaminants that might be there. So it's very important to keep it all clean before it's being used. Excuse me. And then you push the air in through a charcoal filter to make sure that the air going into the vessel is free from contaminants. And then there's another filter which takes the sample, so you can just see it here in this one. So you can use it for collecting from insects. So there's some weevils in here, and I think we were looking for a pheromone with these. So we're collecting the odors from the insects, but it's pushing air through a charcoal filter to filter the air coming in, so that it's clean. And then there's another filter that the air is being pulled through on the way out to collect the sample. So there's some porous material in here called poropac, which is used to trap the volatiles, and leave this running for a period of time, typically a couple of days, and then you come back and collect your sample. So you run some solvent through it to collect a sample of these volatiles. So that's how you get your headspace sample to begin with. But then you want to know, is the insect going to respond to that? So you do a bioassay, and there's various different types of bioassays that can be done. For small insects like aphids, we often use this olfactometer. So this is a walking arena, and you put the insect in the middle, and over a set period of time, then you watch its behaviour, how it's walking around, how much time it's spending in the different regions of this olfactometer, and there'll be one sidearm which has got a filter paper with the test substance, so with the headspace sample that's being collected from the natural odour source. And the other arms will just have a solvent blank, and so we'll be testing to see if they're spending more time, which would be attraction, or less time, which would be repulsion, in the sidearm which has got the test chemicals. So that's olfactometer. You can also do it at a larger scale with these wind tunnels, looking at flying behaviour of insects, and looking at upwind flight towards odour sources, or settlement on targets. So there's all sorts of different bioassays that can be done, but you want to see how is the insect responding to these, and do you get a significant behavioural response, because it's only of interest to us chemical ecologists if it's eliciting a response with the insect, because it could be some really fancy chemicals, but if the insect's not responding to it, then we're not that interested in it really. It's how we're interested in the chemicals because of the effect that it has on the insect. So if we do get something which is behaviourally active, so say we've got something which is strongly attractive or strongly repellent, then we want to know what that is. And the way to do that is to ask the insect. So there's a very cool technique that can be done, which involves recording from the insect antennae. So you actually connect up electrodes with the insect antenna, and you can make a circuit, and then you puff over the antenna the different chemicals. So if you get a complicated natural mixture, often it'll be a number of different compounds. So we use something called a gas chromatograph to separate the chemicals. So you inject the sample in the gas chromatograph, and it separates these chemicals so that they come out over the antenna one at a time. There's effluent coming out from the gas chromatograph column, and it goes over the insect antenna, and then if there's a nice recording, it's a bit fiddly to set these things up actually. If there's a nice recording, you can see a depolarization when the insect is responding to a particular chemical. It won't respond to all of them, there'll be some of them, and that's why this is such an important technique, because it's using the antenna as a biological detector, and we can see which are the critical compounds, because it's not necessarily the ones which are most abundant. The insect is sometimes tuned into some of the things which occur in quite small quantities, but those are the ones which are behaviourally active, and so those are the ones which we are interested in. So we do this technique to see which chemicals to identify, and so that we're only identifying the ones which are relevant for the insect. Otherwise, we're wasting a lot of time identifying chemicals which aren't biologically relevant. So we've done the electrophysiology. We've got some particular compounds of interest. So there'll be a GC peak, and you can use a kind of retention index to locate where that is. And next step is to use the gas chromatography mass spectrometry. So this is a chemical analysis technique, but by